Hey guys, it's Kimberly. How's everyone doing today? I just wanted to say I'm sorry for the lack of videos lately. Being sick will sometimes do that to you. But I did want to come and talk to you guys today a little bit about DNA replication and what goes on behind the scenes when DNA is duplicated during the mitosis of the cell. When the chromosomes come together really and bundle up really close, and then you have the sister chromatids and they break apart and you have two identical daughter cells. Basically, that's the chromosomes are the DNA being replicated, and I wanted to discuss a little bit about what goes on during that process. So, give me just a minute. I'm going to load up my nifty little PowerPoint presentation here, and we'll take it from here. Okay, so here is my nice little PowerPoint slide that I made for you guys. Let's talk about DNA replication and to start that off, what we want to do is we want to talk about what exactly is DNA made of. So, how is it put together? We've got 4 nucleotides: adenine, thymine, cytosine, and guanine. Now, if you look at this picture here, imagine your adenine is red and your thymine is green, your cytosine is blue, and your guanine is yellow. As you can see, red and green is always paired together, and yellow and blue is always paired together. Why? Because adenine always pairs with thymine, and cytosine always pairs with guanine when it comes to DNA. Something else to keep in mind. Just taking a look at this simple structure. It's a double helix. We've got a double-stranded DNA here, a double-stranded DNA sample here. Double-stranded basically just means there's 2 strands. If you look at this picture here, you're going to see one on one side and one on the other. And then the twisting is what gives it the helix shape. These strands run in a 5' to 3' direction. Basically, the 5' end has a 5' phosphate group, and the 3' end has a 3' hydroxyl group. And these strands run anti-parallel to each other. What does anti-parallel mean? Basically, it just means that one strand is going to, if you look at this picture, one strand will say have the 5' end on the top and the 3' end on the bottom, while the other strand runs in the opposite direction. So your 3' end would be on the top and your 5' end would be on the bottom. Anti-parallel just basically means opposite directions here. So, how exactly does DNA replicate? What starts our replication? Let's look at DNA polymerase. This is an enzyme that basically helps to break open the helix with the help of the replication fork. Basically this enzyme is what pairs up the new nucleotides with the old nucleotides. It always, DNA polymerase always runs in a 5' to 3' direction. So, what that means is that it requires a free 3' end to start. So, taking a look at this picture, look at the right-hand side of the picture where you've got your old strand and your new strand at the bottom. Your old strand, that's going to be where your free 3' end is. And the new strand starts on the 5' end. So you've got your DNA polymerase running up this helix towards the replication fork, which is where up at the top where you can see the DNA starting to unzip. That's where the replication fork is. Now, in this case, with the DNA polymerase, you can look at the right-hand side, since that's where we've been focusing on. The right-hand side would kind of be like the leading strand, whereas the left-hand side would be the lagging strand, because the lagging strand always has the 5' end at the bottom, whereas the leading strand will have the 3' end at the bottom. And here we go. Leading strand versus the lagging strand in Okazaki fragments. So, basically what you're looking at here if you take a look at this picture on the right hand side, the top of the helix is where the free 3' end is. So since that's where the free 3' end is, that's considered the leading strand. If you look at the picture, you'll see that the polymerase works in a 5' to 3' direction towards the replication fork, which is at the middle. What this does is this allows the leading strand to be replicated continuously. There's no breaks in it. Unfortunately, the lagging strand has a 5' end at the bottom. Since the DNA polymerase only works in a 5' to 3' direction, it does require that free 3' end. So, in order for it to replicate properly, it has to actually start up at the top where the replication fork is. And it works its way down. Excuse me. In this case, it works its way back to the 5' end. These fragments are because it has to do it in pieces. These fragments are considered our Okazaki fragments, named after the gentleman who discovered them. Once the DNA has been replicated, these fragments are then joined together by our DNA ligase in order to complete the replication process. That's really all that there is to it. There's not a whole lot to replication. This is a very simple description of it. I would definitely recommend, these are my notes here. And in order to kind of get a better idea of the difference between the 5' end and the 3' end and the lagging and the leading strain, I would definitely recommend checking out that bottom link there, researchgate.net. This is an amazing resource. I really do like it. I've been using it myself for a while. Found it last semester working with the E. coli in the lab and stuff like that. So it was really neat. And it's got a lot better answers than sometimes Google does, because it's answered by actual professors and scientists who are working in the field. Alright, so there you have the basics of DNA replication. As you can see, it's not very complicated, but there's definitely a lot going on. Definitely want to look at getting into it a little bit deeper, maybe with the next video, maybe here in a couple of weeks. Excuse me. So if you have any questions, feel free to ask them. I will be more than happy to answer them. You can also take a look at the resources that I've posted below. You can kind of get an idea of where I'm getting my sources, and you can kind of see what's going on. Like I said, definitely check out ResearchGate. It's a great resource. I absolutely love it. And if you like this video, if you want to see more of them, definitely hit that little thumbs up button down there and subscribe, and hopefully I'll get more out to you. Alright, bye.